Hello, everyone, and welcome to this audio podcast. This audio podcast is part of the cytometry cyclone. This audio podcast is part of the Rakas and Bombard episode, and this audio podcast as part of a Storm Robotics production. In this audio podcast, looking at sort of the sort of a subsequent a subsequent or sequential part of the a sort of a subsequent or sequential part of this audio podcast, the part of this audio podcast episode, and looking at now around the importance of interleukin 2, interleukin 7, and interleukin 15, and co-stimulation in context of human kidney transplantation or human renal transplantation. So, transplant immunity results post exposure of a recipient to different different histocompatibility antigens. That is, different histocompatibility histocompatibility antigens in context of serology and the major histocompatibility complex. Transplant antigens are assembled as major so for example HLA and ABO blood groups, and minor histocompatibility antigens. HLA antigens are controlled by a succession of genes of genes or genetics on chromosome 6, referred to when on chromosome 6 in context of human genetics. Human genetics is referred to as human major histocompatibility complex, or MHC. And these genes have been categorized into major identifications. So HLA A and HLA B are examples again of class 1 class 1 genes, and HLA DR and HLA DQ that represent class 2 genes. The genes are also highly polymorphic. HLA-A, for example, has more than 20 alleles, and HLA-B has more than 50 alleles. Laboratory investigations have informed that in human kidney transplantation or renal transplantation, there are alternative sort of pathways that promote the development of a pro-trafficking phenotype of T cells in recipients that have received anti-CD25. Anti-CD25 being a form of a monoclonal antibody in context of pharmacotherapy for recipients who have been transplanted, and at the same time where anti-CD25 sort of represents sort of the older sort of monologues in context of the monoclonal antibody pharmacotherapies. A retrospective hypothesis in amongst literature has been addressed using a T cell culture system that analyzed a T cell culture system to help analyze the effects of anti-CD25 on the expression of specific receptors, and that being a CXCR3 and other important receptors on the surface of T cells. In acute allograft rejection in AAR, in context of kidney transplantation, from the early millennia, there had been an intrigue surrounding some of the most essential interleukins in transplantation. Such as interleukin 2 and interleukin 2 IL-2 and interleukin 15, interleukin 15, IL-15, which had been cited amongst the scientific literature and works, sort of retrospectively. Interleukin 2, IL-2, is also known as a T cell growth factor, or TGF, and it is secreted by stimulated helper T helper cells. So, for example, again CD4, and cytotoxic T cells, i.e., for example, CD8 positive T cells, and large granular lymphocytes. Interleukins at the same time also promote a sort of proliferation of colonial expansion and differentiation of additional CD4 positive cells, B cells, and also activate sort of macrophages and oligodendrocytes. Research has also informed that interleukin 2 and interleukin 15 share several functions. Hence, in amongst the scientific literature, where data or aspects of interleukin 2 have been published, then there will always be aspects in amongst the scientific literature wherein interleukin 15 is also published. So interleukin 15 is distinguished as interleukin 15, distinguishes and sort of prompts an initial stimulation of the proliferation of activated T and B cells, as well as prompting initiation of natural killer cells or NK cells. However, these immune cells also provide complementary assistance to adaptive immune responses mediated by lymphocytes. Interleukin 2 is pivotally implicated in origination of cell reactive T cells, and thereby also facilitates an initiation of tolerance to self. In contrast, interleukin 15 inhibits or precludes this interleukin 2 activation. Interleukin 2 activation induced cell death process. Furthermore, investigations have also informed that interleukin 2 and related cytokine interleukin 15, IL-15, promote maintenance of CD8 positive memory phenotype T cells, whereas interleukin 2 also inhibits or precludes sort of perseverance in vivo investigations. A crucial challenge that remains to be ultimately sort of resolved is to define how cytokines, for example, that share two or more signaling pathway receptor subunits, yet utilize a common signaling pathway that also prompts contrasting functions. That draws this audio podcast to a close. This audio podcast as part of the cytometry cyclone. This audio podcast as part of a Rakas and Bombard episode, and this audio podcast as part of a Storm Lobostics production.